Sorry. Okay, so thank you so much. Good morning, everyone. I would like to thank the organizing committees for this invitation to be here. It's really a pleasure to show some of the work we have been developing. And today I will present mostly unpublished data, so I'm very keen in hearing your comments or criticisms about it. So today I'm going to tell you a little bit about the work we have been developing, trying to understand how nucleus accumbens neurons can represent positive and negative stimuli. So I was really glad that I had the previous presentations because some of the concepts I don't have to talk about them. So when an animal encounters something that is in the environment, it has to decide if it is something that it's either positive or negative, so it puts like a valence stamp to that stimulus. And then it will decide, depending on the experience it has, if it would approach or if it would avoid that stimulus. And throughout the years, we were really interested in trying to understand the behavioral aspects of this mechanism. However, in more recent years, we are trying to understand how this positive and negative information is encoded in the brain, and in specific in the nucleus accumbens circuit. Why nucleus accumbens? Well, there are a lot of evidence from a long time ago, and I really like to show these innate behavior responses, mostly from Barrage's lab, in where they show these oral facial expressions in rodents, primates, and humans that are conserved in response to a positive valence taste, like, for example, something sweet, or to an aversive or bitter taste. And this region has been shown canonically associated with the expression of these oral facial expressions and these liking and disliking behaviors. And the nucleus accumbens is conserved, so it's part of the striatum, is the ventral striatum, and it is conserved between humans and rodents. And it's mostly composed of medium spiny neurons that are segregated into two populations: the ones that express dopamine receptor D1 and the ones that express dopamine receptor D2, although there is a small subset of neurons that express both receptors. And then we also have a smaller percentage of interneurons, but are really, really important. But for today, I'm mostly focusing on the role of these nucleus accumbens neurons in encoding positive and negative information. So, to keep a very simplified version of the circuit, here we have the accumbens, we have the D1 MSNs that project to basal ganglia outputs. This is the VTA. But they also project indirectly through the ventral pallidum that then projects to the VTA, whereas D2 MSNs only projects to the ventral pallidum. And so this is important to keep in mind because I will be manipulating some of these regions later on in the presentation. So do we have evidence that these nucleus accumbens neurons do represent positive and negative information? Well, seminal studies from Regina Carelli's lab have shown that if you give an intraoral delivery of sucrose or quinine, quinine is a bitter tastant and sucrose is sweet, so if you give this to the animals, these nucleus accumbens neurons, they will present excitatory, inhibitory responses, different types of responses, but they will respond very fast to this stimulus. So people started to assume that, yes, well, indeed, these neurons encode something like innate valence to these tastants. And in fact, most of the neurons that they recorded, they only responded to one of the tastants. And if they responded to the two tastants, they would respond in an opposite manner overall. However, these neurons also start to respond to cues that can predict the delivery of something sweet or something bitter. So people also started to assume that these neurons can learn valence because they can attribute then a positive or a negative valence stamp to cues predicting something positive or negative. However, these studies, because D1 and D2 MSNs from the electrophysiological point of view are very similar in extracellular recordings, we still do not know if these are D1 or D2 MSNs or interneurons. And so that's exactly what we were trying to figure out. And so with the development of calcium imaging in specific genetic populations, we expressed GCAMP6F in this case in D1 or D2 Cree animals to selectively target D1 or D2 neurons. We use A2A because D2 receptor is also expressed in a subset of interneurons, and we wanted to avoid those. And then we implanted miniature microscopes, and we imaged the activity of D1 and D2 neurons in positive valence and also negative valence conditions. And in this presentation, I will show you mostly sucrose and shock data, but we also tested other positive and negative stimuli as well. And we did this in different conditions. So we did this when the animals were just exposed to the stimulus with no cue whatsoever, just to sucrose or to shock. And then we also did Pavlovian associations. Classical, you have a cue, you have a reward, or you have a cue, and you have an electric shock in the paw. And then we also tested in extinction conditions in where animals still hear the cue, but they no longer have the sucrose or the shock. Okay, so what we found in the beginning, this is just animals exposed to sucrose or shock, and as you can see, in blue, you have neurons that only respond to sucrose, in red, neurons that only respond to shock, and in yellow that respond to both stimulus. And in fact, what you can see is that you have all types of neurons, and this is D1 and D2 data. And what I would like you to see is that it's very similar, the type of responses we have within D1 and also within D2 neurons. And recapitulating the electrophysiological studies of non-identified neurons, we also observed that sucrose elicited mostly inhibitory responses, here in blue, whereas shock elicited mostly excitatory responses, both in D1 and in D2 neurons. So this basically recapitulates what people have shown before, using other tools. So, but what happens if you look, when you dig more into the data and you look trial by trial response of that neuron? And here I have to assume I was very naive when I started to do this, because I was assuming that a lot of neurons in the accumbens will preserve their response to that stimulus. So if they would have had an excitation in the first trial, I was assuming that in the subsequent trials, they will also have an excitation to that stimulus. So that was my assumption. It was wrong. Because when we look at, this is just different neurons, we have way more neurons than these, but if you look throughout the trials, you see that basically they increase, decrease, or no change in activity when they are getting sucrose, and we saw the same for shock. We did other bunch of analysis with the correlation of activity. We also did Shannon's entropy that measures the variability in neuronal activity, and basically all the measures shows that there is drift between trials in the same session. So the neuron does not respond always in the same manner. So then we went to do machine learning, and we did also for single neuron, but I'm showing you population-based, because if you have this individual drift, the way that this brain region might be representing this information might be at the population level. And so we trained a decoder to see if we could distinguish US1 or from US2 trials. And as you can see here, the activity of D1 neurons or D2 neurons could be used to accurately classify a sucrose or a shock trial. Okay, so you have this representation of positive and negative stimulus at a population level despite this individual change in responses. So then, next, we did the Pavlovian conditioning, and this is where the animals have a cue and they have the sound, and as you can see, you don't have to look at the percentages or anything, but if you look at this is very similar between, again, D1 and D2 neurons, and for us, this was really surprising. I was expecting to see some changes, some differences between these neurons. And when you look throughout the days of the, throughout the days of the training of the Pavlovian condition, day 1, day 5, and day 10, and if you combine the different types of responses, there is also some stability at the population level. And again, if you look at each neuron, and now we have way more cells, but if you look at each neuron on day 1 and the type of response it has, For most of the neurons, this response is not conserved in subsequent days. So again, we show that there is this drift in activity. And if you do cross-correlation of that neuron response throughout the days, it's also that it's not correlated whatsoever. So there is a stability in this representation at the population level, and there is individual variability. But then we thought, well, maybe we are looking at all neurons. What about if you just take the best neurons, those that reliably encode information on day one, and what about if we follow those specific neurons throughout the days? And that's what we did here. So we took these top 20 D1 MSNs or D2 MSNs, and we checked for the accuracy in identifying a Q, or for example, the sucrose throughout day 5 and day 10. And as you can see, while on day 1 these neurons were really had good decoding accuracies, on day 5 or on day 10, they no longer presented this. And as you can see here, so these top 20 cells throughout the days, they basically overlap with the other population, showing again that what you do in the first day does not necessarily mean that you will do the same in the subsequent days. And again, we saw that at the population level, if you look at the population, you still have representation, good representation of what is a Q, what is sucrose, or what. And we also found this, and I'm not going into detail, but we also found this for aversive cues and aversive stimulus. So it's basically the same. Okay. So the population represents this. We can distinguish between a Q and something positive. We can also distinguish between a Q and something aversive. Can we distinguish the two Qs of opposing valence? And basically, so we did neuronal trajectories trying to identify the dynamics in activity. And what we saw, and in blue you have the trajectory of the positive valence Q for sucrose, and this is, in red, is the negative valence Q for the trajectory for the negative valence Q, and you see that they go in opposite directions for both D1 and D2 MSNs, meaning that this information is differentially represented in nucleus accumbens D1 and D2 neurons. Okay, so our next question was, if we modulate the activity of these neurons, because they are so important, they appear to be important for these Pavlovian associations, can we alter the responses, the conditioned responses? And so what we did, we performed optogenetics, activating or inhibiting D1 or D2 neurons, and we did this activation during the Q period in both positive and negative Pavlovian conditioning. And to keep a very long story short, in the appetitive Pavlovian conditioning, what we found was that regardless of what you do, if you activate or inhibit D1s, or if you activate or inhibit D2 neurons during the Q period, you change the conditioned responses. And in fact, you abolish, or you delay, I would say, the association. So this led us to speculate that, and we didn't have any impact in the aversive, so this was only for appetitive. But this was not so surprising, because association of the shock with the Q is a quite powerful association, and we didn't have any impact of manipulating these neurons. So this led us to think that you need both populations having this coordinated activity in order for these associations to take place. So next we tried to understand what was the role of these neurons in extinction conditions. And what we did was to expose the animals to the same cue that they learned, but then now you don't give sucrose, and you also don't give the electric shock. And what you can see, this is appetitive extinction. You see that the animals, they change their behavior when you are doing 3 days of extinction. And again, when you look at the activity of D1 or D2 neurons throughout, so this is the last conditioning day, extinction day 1 and 3, basically these neurons, D1 and D2, they were remarkably similar in the terms of overall population responses. So far, we were a little bit surprised because D1 and D2 neurons, they appeared to be so similar in everything that we were registering. The activity was really, really like mirroring each other. But the thing is that when we perform the extinction of aversive, in aversive conditions, so now the animal hears the cue, but you don't no longer see the shock, and we analyzed the extinction day 1 and 9, then things got different between D1 and D2 MSNs. So this is the PS stages of the activity, increases, no change neurons, and inhibition neurons. But what I think is more interesting is that when you look at the responses that you have on the last conditioning day, and you compare that to the responses of that same neuron on extinction day, you can see that in D1 neurons, most of the excited neurons on day 1 are inhibited on extinction day 9. And most of the inhibited neurons on day 1 of conditioning are still inhibited on day 9 of extinction. However, if you look at D2 MSNs, this is clearly different. So most of the excited neurons in response to the cue on day 1 on the day of conditioning then present excitation on the extinction day. And the inhibited neurons, most of them also present this excitation. So it's clearly different between D1 and D2, the way they are representing this extinction association. And so what we did next was to manipulate these neurons during this cue in extinction conditions to see if we could change animals' behavior. And so what you have here is the freezing responses. If you manipulate D1 neurons, you do not change freezing responses. So we did activation and inhibition of these neurons in extinction conditions. However, if you do that with D2 neurons, if you inhibit these D2 neurons that I showed you that they were hyperexcited in extinction conditions, then we are able to delay the extinction of the conditioned response. So this led us to believe that these D2 neurons are really, really important for this extinction process of aversive CS-US associations. Okay, so up to here I showed you Pavlovian associations, and one other question that we had in the lab was, what about classical positive and negative reinforcement tests? And a very talented postdoc in the team, Karina, so she did a very simple experiment in basically where she used the classical conditional place preference test, and she optogenetically activate D1 or D2 neurons in one side of the chamber to see if the animals would manifest some preference or some avoidance to that chamber. And what she found was quite surprising at that time. This data is published, so I'm not going to take too much into that, but what she found was that depending on the type of activity you trigger in nucleus accumbens neurons, this is D1 in green and D2s in blue, you can have both place preference and you can also have both place aversion in the same individual. So this is dependent on how much you activate these nucleus accumbens neurons. And so why is this happening? And one possibility was that if you activate these neurons in a more prolonged manner, maybe you are releasing endogenous opioids in downstream target regions. So MSNs are classical GABAergic neurons, but they are also able to release endogenous opioids. And D1 MSNs, they release dynorphin, and D2 MSNs, they release enkephalin. And endogenous opioids, as you might know, they are really, really important for rewarding and aversive responses. And so our best hypothesis was that these opioids were being released in the pattern 2 of activation of these neurons. And so what we did, and this is published data, so I'm not going to take too much into that, but summary, if we use antagonists in the VTA when we stimulate D1 MSNs, or if we use antagonists of enkephalin receptor in the ventral pallidum, we are able to abolish the place aversion that we were observing when we were activating nucleus accumbens neurons. So these mean to us that these endogenous opioids were being released in these regions and that they were important for the behavior that we were observing. So the question was, when we are manipulating the system, right, and we are releasing these opioids, but we asked ourselves, maybe these opioids are released in vivo, in freely behaving animals, but when is that happening? And in fact, we don't know because it's technically very challenging to measure these opioid molecules in vivo. And so we teamed up with Lin Tian from UC Davis. So Lin developed the D-Light fluorescent indicator for fluorescent sensor for dopamine. And Lin and her team developed a sensor for dynorphin, which is named K-Light 1.3, and also a sensor for enkephalin, which is named Delta Light. And so this is just cell validation, and basically it's very similar to D-Light, but it's just using the dynorphin receptor molecule or the enkephalin receptor molecule. So what we did, we injected the K-Light in the VTA because as I showed you before, D1 MSNs can release dynorphin in the VTA. So we injected this sensor there, and first we did the pharmacological validation. So we used an agonist of the kappa opioid receptor, which is the dynorphin receptor. We saw this very nice increase in activity. And then we tested the animals in both, again, Pavlovian associations, both positive and negative. And so what we found was that, and this is very preliminary data, so I mean, we have more data supporting this, but this is in the VTA, and basically we saw dynorphin transients both in appetitive and also aversive conditions. And this is quite cool because for the first time we can see these molecules in vivo. Okay, so to sum up, I showed to you that D1 and D2 neurons are recruited during positive and aversive stimulus, that also you have mostly a population representation of this stimulus. Dynorphin is released in the VTA both in positive and negative experiences. And also, if we manipulate these neurons during Pavlovian conditioning, we are able to change the conditioned responses. And one thing that it was really interesting is that these D2 neurons, but not so much D1, are important for extinction of aversive conditioned responses. And now the most important slide, so the people that mostly contributed to the work, also the wonderful collaborators and the funding agencies. And I'm glad to take any question. Thank you.